Thank you, ladies and gentlemen. Today we are going to take the introductory part of microbiology. Microbiology is the study of tiny microorganisms that cannot be seen with the naked eyes, and most importantly, they include the bacteria, viruses, the fungi, and the protozoans. Bacteria are prokaryotic organisms which can be gram-positive and negative. They are capable of both causing infection. They have their industrial use and they are found in the microbiota of the human body and other related hosts. And these microorganisms can be strategically classified based on their metabolic activity and their biochemical characteristics, which we will take in the further classes. They have their respective characteristics which can be used in differentiation and classification of the organism, most importantly in understanding what they are. First of all, they are small in nature. They range between 0.01 micrometer to 0.02 nanometer. Some of them they are in centimeter like Thiomigrata namibiensis, and they do not have membrane-bound nucleus such as the Golgi apparatus and the rough endoplasmic reticulum. They consist of the DNA in their chromosome, which is located as genetic bacteria in the nucleus. You can see the ribosome is used for the synthesis of protein. The cytoplasm contains the organelles, the chromosome, the DNA. The cell wall houses the entire cell. The cell membrane contains the protein-bound nucleus for transportation. The flagella is for movement and locomotion, and it is composed of what is called the flagellin. Here you can see the cilia around the bacteria itself, and you can see some tiny dot. They are they are both lysosome, which they are responsible for digestion of food substances. Then we have the fungi. Fungi are multicellular organism. They are eukaryotics. They contain the membrane-bound nucleus, and they are filamentous in nature. They are composed of what is called the cellulose and the chitin. They have the high capability of utilizing, or let me say, metabolizing lipid and glycogen. Most importantly, they can only dwell where there is water or moisture, and they are one of the most prevalent organisms, as everywhere they are you can see them in the air, and they are important in both industry and medical microbiology. Yeah, this is depiction of typical yeast cell. You can see the mitochondria around and vacuoles, which are entirely absent in prokaryotics. They share the same characteristics with prokaryotics. However, they are distinct in their metabolic processes. You can see there is the nucleus, and the nucleus is bounded. Now, viruses. These viruses are obligate intracellular parasites. They are filterable. That means their nuclear protein containing DNA and the RNA can be filtrated and analyzed for further analysis. They are crystallizable, meaning they can live in a dormant stage outside the body for a long period of time. Immediately they enter into the host, they can use the machinery of the host to produce the daughter or viral particles. They don't have metabolic processes entirely. They hijack the metabolic processes of the host to produce particles of the viruses and subsequently infect the virus through what is called the tissue trophism, confirmatory aspect of the virus. Here you can see the spikes. The pole in the spike is responsible for the attachment. And the protozoans, these are mainly parasite, and they are unicellular and microscopic. They have a symmetrical morphology. Most of them they have symmetrical morphology. Example is the amoeba, the euglena, the plasmodium, and the category of helminthes, which are mostly very dangerous parasite, though opportunistic. This is yes, sorry, it's a repetition of what we already did, but you can recap from what we discussed recently. Yes, this is very important, as you know. It's very good in the fermentation of carbohydrates and advanced processes. Now bacteria. We know when we say bacteria, you are very conversant with the differentiation of bacteria into gram-positive and negative. Likewise, viruses are classified into double-stranded and single-stranded RNA, while fungi are divided into mold and yeast, and some certain unicellular bacteria that they are important in causing diseases and production of antibiotics. Now, the domain of microorganisms. They are categorically classified, as I have said, the prokaryote and the eukaryote. The prokaryote contain the bacteria and the old version of the bacteria, the archaea bacteria, while the eukaryote consists of the cells, unicellular and both multicellular organism carries the eukaryotic cell. The protistas, the fungi, the plantae, and the animalia, they consist the advanced cell or the complex cell. That's why they are under eukaryotic cell, and what differentiates them is membrane-bound organism. Now, example of this bacteria we have discussed is the Staphylococcus aureus. And Staph aureus are gram-positive. They are named as Staphylococcus aureus because of the stratified arrangement of the cocci. That's why they are called that. That's why we say the Staphylo stands for the branches of the cocci. Now, there are other features that they are very important in the classification of the bacteria. The bacilli technically mean the rods. The spirochete mean the coiled morphological structure of bacteria. Diplo mean two cocci, and cocobacilli, arrangement of the two cocci to give one nature of a rod shape, which is called a bacilli. Then the vibro, most especially, it means example of these vibros are the vibro cholerae, Staphylococcus aureus. We have already discussed that, and this is a defection of bacteria. Recently, we say genetic material. This is just material stands for DNA, and in some certain organisms like the viruses and RNA, and it contains the central processing unit of the bacteria. Here, this is the morphological demonstration of the cell structure, cell wall, and permeable membrane of the bacteria. Specifically, this category of the bacteria, section of the bacteria, is used in the differentiation of the bacteria into gram-positive and negative. Here in the lower portion, we have the permeable membrane. The blue contains the peptoglycan layer, which is used in differentiating the bacteria into gram-positive and negative. The peptoglycan layer takes the color of the crystal value, which is blue, as the first dye, while the gram-negative bacteria take the color of the safranin. This is because of the thickness and thinness of the peptoglycan layer in gram-positive organism, and the thinness of the peptoglycan layer in the gram-negative organism. Is the fiction, lower layer, permeable membrane, peptoglycan layer, phospholipid layer, phosphoprotein layer, and then in the inner lipopolysaccharide layer. The lipopolysaccharide layer consists of what is called the A antigen, the copolysaccharide, and the lipid A,